Boa tarde a todos. É um imenso prazer recebê-los em nosso canal do YouTube. Eu, professor Daniel Bruschi, em nome de toda a nossa equipe, dou as boas-vindas a todos. Com certeza é um momento bastante aguardado por nós neste ano. Inauguramos este canal para manter toda a comunidade científica de pesquisadores, estudantes conectados ao longo de todo este ano de 2021, enquanto aguardamos o nosso evento presencial que ocorrerá durante o ano de 2022. Esse canal, né, vai receber pesquisadores nacionais e internacionais com o intuito de servir como um repositório mesmo de conteúdo de qualidade para mantermos informados e muito atualizados em temas contemporâneos, citogenética e citogenômica. Então, a cada dia, né, a cada 15 dias, um novo tema será abordado, e a gente convida que todos possam participar ao longo de todo este ano de nossas interações. Qualquer interação que a gente tiver com o palestrante, ela vai ser realizada pelo meio do chat. Perguntas e comentários poderão ser publicados aqui no chat, então, e nós transmitiremos na medida do possível e orientaremos as palestras a partir daí. Novamente, eu agradeço a presença de todos, peço a permissão de vocês para que a gente possa iniciar nossa tão aguardada primeira live. Let's start your first lecture titled Heterocromatin, Structure and Function. I have a great pleasure to invite Dr. Marta Svartman from University of Minas Gerais and Dr. Aparecido da Cruz Peixoto from Pontifícia Universidade Católica de Goiás that co-share the sections with me, Dr. Daniel Bruschi from University of Paraná. Thank you so much for your presence here, and please, Marta, have the honor to invite and introduce your speaker, Dr. Ivan Eicher.
So good afternoon. Thanks a lot. I am super pleased to introduce Dr. Professor Evan Eichler to give our first talk. Dr. Eichler is a professor of genome sciences at the University of Washington, and he's also an investigator at the Howard Hughes Medical Institute. He is a fellow of the National Academy of Sciences in the US, and he has hundreds of very nice and important pieces of work. His work has focused on genome, structure, function, and evolution, especially on primates. So I want to thank him very, very much for so graciously accepting our invitation, and remind you to write on the chat all your comments and questions in English or in Portuguese, and with that we can finally start our first lecture. Thanks, Dr. Eichler.
Thank you, Dr. Svartman, and thank you, Dr. Bruschi, for the introduction. I'm going to share my screen now, and let's just hold for a second here till I get this up. So can everyone see my screen? I'm going to shut this off. Okay, well, it's a pleasure to be here today. I'm going to talk to you today about some of the new insights we've gotten over the last few years in terms of the evolution, the structure, and complete sequence of the human genome. And I'm going to start really with a subtext, that although I'll end on a discussion with respect to heterochromatin, its evolution, its structure, and its function, I'm going to start really more broadly what I think is a new revolution for human genetics, which is the ability to do long-read sequencing of human genomes. So the challenge for sequencing the human genome or characterizing it is that human genomes are complex. So what I'm showing you in this relatively messy diagram are all the segmental duplications, pairwise relationships between duplicated sequences that exist between human chromosomes. So wherever you see a red or a blue line, that means there is a piece of DNA that's 20 kilobases in size and 95% identical at multiple locations. So you see that areas near centromeres, which are indicated here in areas near the ends of chromosomes, are particularly enriched. So why is this important for a cytogeneticist to understand? Well, the importance is that about 50% of all large-scale copy number variation that's greater than the kilobases, so differences between different humans, map to this 5 to 4% of the human genome. We know from work from our lab and Jim Lupsky that the presence of these large duplicated sequences create genomic hotspots that recurrently delete and duplicate in association with autism. And last, which I won't talk much about today, we also know that there are genes in this duplicated material. These genes in particular, recently there's about a half a dozen of them that have been shown to be specific to humans, different from other non-human apes, and important for actually the expansion of the frontal cortex. The problem is that these duplicated regions, by dint of the fact that they're highly identical, have been very, very difficult to sequence resolve. And if we extend this to the purple, which are the centromeres and the acrocentric regions of the human genome, we have a large fraction of very dynamic parts of our genome that have not been sequence resolved. So the solution to this really has been long-read sequencing. And I'm sure many of you are familiar or have heard of this new technology that has really taken hold in the last 5 years. There are two main companies that are generating or providing instruments for this type of work. One is Pacific Biosciences, which actually sequences native DNA molecules anywhere from 15 to about 50 kilobases in size. And the way it does that, it actually has its DNA polymerase localized at the bottom of a well, which is called the zero-mode waveguide. And fluorescently labeled dNTPs get incorporated, lasers excite, and you can directly visualize the incorporation of nucleotides into long DNA molecules. The other technology, which is shown here on the right, is developed by Oxford Nanopore Technology. It has a higher rate than the PacBio sequencing technology, but it detects sequence differences in a completely different way by looking at ion potential across a membrane, and based on the actual change in voltage or change in current as this comes across, you can actually infer the base pairs that are being sequenced. It has the advantage of being able to actually thread through this extremely long DNA molecules, in some cases over a megabase in length. In the last 2 years, there have been two important advances in both of these technologies. The one, which is so important now to assembling human genomes, has been the ability to generate high quality, highly accurate sequence. So the way it's done is that you take DNA molecules, you put these smart bell adapters on them, and then the polymerase, which is now very highly processive, actually goes through the actual sequence molecule, then through the hairpin adapter, comes out the other end and continues to sequence around the molecule. And so while there's a high error rate on any individual subread, when you get enough let's say 4 or 5 of these passes through the actual sequence itself, you can generate a consensus that is highly accurate, essentially 99.9% accurate. The other thing with respect to Oxford nanopore technology, which has really shown to be quite useful for assembling genomes and including heterochromatin regions, has the ability to generate ultra-long reads. So what are ultra-long reads? So when we're talking about ultra-long reads, we typically refer to sequence reads that are greater than 80 kilobases in size. And so this is showing you data on a size distribution, and this is a log scale, of the number of sequence reads we generated from a human genome, where the contig N50 is 139.9 kilobases. That means that 50% of the sequence reads corresponding to a genome size, a human genome size, are in sequence reads that are greater than 140 kilobases in length. This is so powerful because this allows us to traverse very complex regions of the genome. So, in terms of this technology, there's a couple things I want to discuss briefly in terms of the advances. The first has been the ability to actually detect and sequence structural variation much more comprehensively than has been done previously. So this is work from a postdoc of mine, Mark Chasen. And when we started this project, I don't know, 5 or 6 years ago to sequence the first human genome, this first human genome was incredibly expensive. It cost us about $70,000 to generate the sequence reads. This is from PacBio. But we selected a human genome that was essentially a Heterocromatin mole. What does that mean? That means it actually only has one complement of the 2 potential parental complements. So for those of you who don't know, a complete Heterocromatin mole is formed when a sperm is essentially fertilizes an egg, but the female component or the maternal component is essentially lost. So this essentially fertilized egg actually diplotizes the paternal complement, and this creates essentially a haploid genome, which only has the paternal complement. So the reason we selected this as our target for generating the first maps of structural variation and then the first assemblies is that we didn't have to consider 2 genomes at the same time, both the maternal and paternal. We just wanted to focus on essentially one haplotype to characterize. So Mark developed an approach, basically identify structural variation based on signatures of mapping these long reads back to a human genome. Once you identified the reads, you then assembled them. Once you assemble them, you compare them to the reference, and you could sequence resolve the breakpoints. And the really important conclusion from this work was that, in fact, when we compared the genome that we had sequenced with essentially data that we had generated from the same genome with Illumina, so this was standard next generation sequencing technology, we discovered that more than 75% of all the structural variation that we discovered was essentially novel, i.e. had not been detected by next generation sequencing technology. And so this is just showing you a distribution of all the events that were discovered. So these are just insertions in black or gray with respect to the reference, deletions, which are either red or pink with respect to the reference. And whenever you see dark black or dark red, that means that essentially the variation was essentially newly discovered as a result of long read sequencing. So 92% of the insertions and 60% of the deletions were essentially novel. And while you would get from a given genome typically 3 to 4 million single nucleotide variants, these 22,000 novel structural variant events correspond to about 11 megabases of sequence that's present that now could be accessed and studied. So the other really important application of this technology was to essentially finish the human genome. And it may be apropos that this is the 20-year anniversary of the sequencing, or at least the first publication of the human genome. And what I'm showing you now is a new reference genome, which we call T to T. So this is telomere to telomere, where we targeted once again a complete Heterocromatin mole. And then we sequenced and assembled essentially entire chromosomes from end to end. And I'll explain a little bit later how this was achieved. But just to let you know that essentially every chromosome that's shown here with a solid gray line means that we now have telomere to telomere assembly from P to Q of each of these chromosomes. There are still regions that are shown here in white. These represent acrocentric or parts of the acrocentric region of the genome that as of like 5 months ago were not resolved. But as of actually last week, these regions are now essentially fully resolved, at least for one human genome for the first time. So when you have this information, you can go back and ask questions regarding how the genome is organized. And this is our own analysis that we did essentially a few weeks ago, which shows that essentially the proportion of the human genome that is actually duplicated has increased from 5 to 7%. And so we now have all this additional structural variation and potential variation in terms of segmental duplication that we previously could not access, we now can see. In addition, we now have sequenced and assembled human centromeres for the first time from one reference human genome. So I'm going to talk more about this today, but this is showing you essentially chromosome 8 centromere going from the P arm all the way to the Q arm. And the only thing I want you to notice right now is what this graph underneath represents. So for those of you who are unfamiliar, the centromere is thought to be composed of 171 base pair motif or repeats that are repeated over and over again into higher order structures that are actually then they themselves are repeated. And what I'm showing you in this colorful heat map is basically taking this 2 megabase sequence assembly of chromosome 8 and breaking it into 5 kb segments and comparing it to every other 5 kb segment that's present in this sequence. So whenever you see red, that means that that sequence is high sequence identity, close to 100%. And as you move away, you can see that these in fact become different colored. As you get out to the real periphery, you can see that the centromere is made up of what appears to be much more divergent sequences. But interestingly, you'll notice that these two satellite paracentromeric sequences are much more identical to each other than they are anywhere else in the genome. So this particular segment is highly identical to this identical segment shown on the periphery, suggesting that the centromere has evolved in layers. The other thing we've been able to do is get our first glimpse of what the acrocentric sequence looks like. So these are the 5 acrocentric short arms that are depicted here. Each line here represents essentially one of the acrocentrics. These are the gaps that I mentioned before that have now been recently filled. These are composed of rDNA sequences that are repeated over and over again. In this particular hydatidiform mold, there are about close to about 300 such rDNA units. And what I'm indicating here in red is perhaps a surprise. It turns out that when we look at the acrocentric regions, we find out that they're composed of more segmental duplications than anything else. So when we compare them to satellite sequences, where we compare them to rDNA, or even the centromeres, the biggest constituent of acrocentric chromosomes is, in fact, the segmental duplications. So in addition to the rDNA, in addition to the centromere and other forms of satellite, there are these essentially large blocks of segmental duplication that are shared both between acrocentrics or among acrocentrics, but also to other chromosomes. So the really exciting thing is that once you have the complete sequence, you can now design probes specifically to recognize specific parts of the acrocentric. So here's a probe, for example, that will recognize chromosome 13, 14, and 21. And you can design probes throughout the entire region to begin to interrogate the acrocentric in this cell line as well as other human cell lines. So in this slide, what I'm showing you is some work from my colleague Mario Ventura in Italy, where we've designed probes. So shown here on the left is a probe that was designed to target segmental duplications on chromosomes 13, 14, and 15. We tested 3 different humans. So this is the original cell line, hydatidiform mole. This is a Puerto Rican sample. This is another human sample. And you can see that this particular variant that we detected, this probe, is essentially fixed in the sense that we see it on every human chromosome 13, 14, and 15, but in all 3 humans. Here's another probe that we decided that went to chromosome 13, 14, and also the paracentromeric region of chromosome 18, a non-acrocentric. And I think you can clearly see as you compare other human cell lines that not every chromosome and every human actually carries this. So this is not new to cytogenomics. People have known this for years. These are called heteromorphisms, but we now can systematically characterize the heteromorphic and fixed structure of acrocentrics because we have the first reference sequence for the acrocentric chromosomes. So the next challenge is moving away from heterotitiform molds and being able to actually generate fully phased and assembled diploid human genomes. And this is work that's ongoing and is an active area of research by many labs. But I want to simplify by talking about basically a simple strategy that has been put forward and is now being essentially applied in labs across the world. The idea is if you want to sequence and assemble a human genome, you got to distinguish both the maternal and paternal haplotypes. So the formula that people have actually recently published on is to generate high quality, long read, high fidelity sequence, take that human genome and create an initial assembly where the contigs are not particularly phased, identify all the SNPs, both heterozygous and homozygous that are present, and then to couple it with linking or phasing information. This can be done from high-C, strand-seq, or even from parent-child trio information that you may have from NGS. Once you have that, you have this essentially this map of the SNPs, and you basically identify all the SNPs and you phase them so you can actually distinguish the paternal from the maternal complement. And once you've done that, you go back to the underlying raw reads and you then partition the reads into whether they're maternal or paternal. And then you assemble not one human genome, but actually two human genomes, one for the mother and one for the father. So this was recently published, and this is to show our first assembly of a diploid human genome. It's not a 3 gigabase genome anymore. It's actually a 6 gigabase. And so when we assembled it using this strategy, using essentially linking information from strand-seq coupled to hi-fi sequencing, we assembled two haplotypes. And I'm showing you here for convenience the paternal in blue and the maternal in pink. And you can see that we've actually assembled most of the human genome, about 95% of it, into contigs of 25 to 28 megabases in size. There are still some areas that we haven't resolved in a diploid sense. So this includes the acrocentrics, some of the centromeres, and even some of the pericentromeric segmental duplications. But overall, we've assembled with an accuracy of close to about 98% the maternal and the paternal haplotype. If you look very carefully, you'll see areas where sometimes you see a blue juxtaposed to a pink. These represent potential switch errors, areas that we actually put the wrong haplotype in the wrong location in the genome. We estimate the error in terms of phasing at about 0.2%. This, I think, is revolutionary because it's going to be, I think, the way we're going to think about analyzing human genomes in the future. No longer by simply aligning reads to a reference, trying to infer local structure, but actually to build genomes assemblies first and then characterize the variations second. And so that's exactly what we did with this Puerto Rican sample. We basically characterized all the structural variation. So this is a heat map showing you roughly the 25,000 structural variants that were identified. And wherever you see this kind of spike, you see it near the ends of the chromosomes. This is a property we've known for quite some time. The structural variations increased probably by a factor of about 4 to 5-fold near the ends of human chromosomes. So this Puerto Rican sample, HG00733, contains about 25,000 structural variants that we now have fully sequence resolved on maternal versus paternal haplotype. And for those of you who are using next generation sequencing, I think an important point is that about 75% of this variation is missed by strictly applying Illumina WGS or whole genome shotgun sequence. The cool thing is that in addition to the structural variants, we also have the insertion deletion events, and we also have the SNPs. So we also have a complete SNP map showing you the locations of all the structural variants in the context of their flanking SNPs. We can fully sequence resolve loci like HLA, and we can actually assign the SNPs to the structural variants in conjunction for future genotyping. So based on this work, a team of us. This is roughly about a half a dozen investigators across the world, decided to actually build the first, really a panel of long-read sequenced human genomes that were fully phased and assembled. And so we went back to the 1,000 Genomes Project. Some of you may be familiar with this. This was a sampling of human diversity. We selected one genome from each of these populations. So we selected 32 reference genomes from the 1,000 Genomes Project, and we applied essentially this phased sequence and assembly-based strategy to characterize it. And I want to highlight here two young researchers, Peter O'Donnell and Peter Ebert, who really led this charge. Peter Ebert from Germany and Peter O'Donnell when he was at my lab. So what we successfully did was we phased and assembled 64 human genomes. Remember, there are 32 individuals, 2 haplotypes per individual. And shown here are the roughly 110,000 structural variants. This is a cumulative plot showing you how many new structural variants we're detecting in normal human genomes for every genome that we phase and assemble, for every haplotype we phase and assemble. So the colors here, shown here on the left, indicate two things. So one of the things I want to highlight is this red line here. The red line represents essentially structural variants that were seen in every human haplotype that was sequenced that was not present in a human reference genome. So these likely represent errors. There's about 1,500 of them, where we know that every other human that we've sequenced and assembled, their haplotype differs from the human reference. This purple represents essentially structural variants that are seen in the majority of humans, but are not present in the reference genome. So the reference genome carries the minor allele. And then the blue represents polymorphic, and the light blue represents singleton events. The other thing that you'll see here is that I've broken out, this is a cumulative plot as you go up. So this is the addition of every new human haplotype. You can see that we're continuing to add about 1,000 to 2,000 new structural variants. So we need to probably sequence hundreds of human genomes to saturate this. But you can see something that we would predict, African samples compared to non-African samples. Africans, even for structural variation, show much greater genetic diversity. So you're getting an extra 500 to 700 structural variants that are new if you actually sequence an African compared to sequencing another non-African sample. Now, the really important thing, I think, for the human genetics community is once we sequence resolve the structural variant, once we know the SNPs that are in complete linkage disequilibrium, we can now take these sequence resolved structural variants, go back to the Illumina data. So this is now going back to Illumina data from 3,000 human genomes on the right. And we can genotype accurately, we believe, about 50% of all the structural variants that we have sequence resolved. And so this is just showing you the genotyping results from 50,000 structural variants that we fully sequence resolved. So maybe what's relevant to some of you here, because I believe there are talks from basically using Illumina whole genome sequencing, which is often called next generation sequencing, versus BioNano. When we did these studies, and starting back even in 2019, we did all of the samples with all 3 technologies. In other words, we generated the same genomes with Illumina WGS, the same genomes with BioNano, the same genomes with PacBio sequencing. And what I'm showing you here are just 3 human genomes for comparison. It doesn't really matter which the genomes are, but these are 3 of the 36 or 32 genomes that we characterize. And I'm showing you the relative gain from long-read sequence and assembly, which is shown in blue, versus next generation, or what we call short read, which is shown here in red, versus BioNano genomics, which is shown here as optical mapping in black. So insertions are shown to the right, deletions are shown to the left of this line. And just to point out that the count of structural variants is shown here as a logarithmic plot. So this difference here represents a really sizable difference in terms of increased sensitivity. So just to kind of keep this in the back of your mind, when you sequence a genome with Illumina, in terms of detecting structural variants, you'll detect roughly, if you apply multiple colors, about 11,000 structural variants. But with PacBio, we've shown multiple times, somewhere between 25 to 30,000 structural variants. So in other words, 75% of the structural variation is being missed. Shown here is bio-nanogenomics detection. And you can see here that there's almost no detection of bio-nanogenomics below 10 to 5 kilobases. In fact, we've determined that 95% of the structural variants are actually missed completely by applying a bio-nanogenomics. However, bio-nanogenomics does detect structural variation events that are larger, that are being missed both by Illumina as well as by PacBio. These are mapping preferentially into heterochromatic regions, preferentially mapping to regions of segmental duplication. And we believe these are actually still not fully resolved, but we have a strategy for resolving these in the future. So here's one of those regions that we discovered by actually combining long read as well as bio-nanogenomics. This is showing a region of the human genome that's about a megabase in size. What I'm depicting here in these different colors is I'm depicting inversions, duplications, deletions that are present in different normal human haplotypes. So it's an area which is indicated along the top that is rich in duplicated sequences, but it's also rich, as you can see from this browser snapshot, in genes. This region was not fully resolved by our PacBio sequencing strategy, but bio-nanogenomics detected essentially this type of variation very systematically. So there's we estimate about 1,000 such regions that are present in the genome. 75% of these are supported by copy number variation analysis with Illumina next generation sequencing, and about 50% of these localized to seg dupes and overlap with genes. So in the end here, I want to talk about some of our most recent work and how we're going to resolve these more complex regions of the human genome, including large blocks of segmental duplications like I just showed you, and areas that are typically associated with centromeres and heterochromatic regions, and what we can learn when we get the sequence. So our strategy to resolving these regions is essentially to combine both the accuracy of hi-fi sequencing reads with essentially the ultra-long sequencing technologies that are being developed by Oxford Nanopore Technology. So shown here is what we now do for a given genome. We generate ultra-long sequencing data, which gives us these long sequence reads that are over 100 kilobases in size. And we also generate high quality, high accuracy, hi-fi sequencing data. We build an initial assembly with the hi-fi data, and we use that data to identify sequence contigs, both from the maternal and paternal. And then we look for areas, base pair changes, we call them single unique k-mers, or SUNCs for short, that exist once and only once in the genome. And we take those from the hi-fi data and we encode the ultra-long reads. So shown here is a barcode for the ultra-long reads, where the SUNCs are indicated here in color. We then take these ultra-long read barcodes and we use them to scaffold across complex regions, like the centromeres, like the large blocks of segmental duplication. Once we confirm the overlap of these ultra-long reads from the SUNCs that came from the hi-fi data, we then fill in the sequence from the sequence contigs that were generated from the hi-fi data. So this is essentially how we sequenced and assembled the first human genome, was with this type of strategy. And I'll just show you some of the work that'll come out this week. Work primarily from Glenys Logston to show you the sequence structure and organization of the chromosome 8 centromere. So I already showed you this image before, but shown here is the structure, once again, of the centromere, chromosome 2. And you can see there's 5 kilobase segments we've been compared to each other. And I'm showing you here in red, once again, the areas that are virtually identical to one another. So this particular segment of these 2 megabases of the centromere is essentially 100% identical. And as you move away from it, both left and the right, you can see that it's actually getting more divergent. So you can see this is breakdown in terms of the colors. But if you notice and you look very carefully, you can actually see that there are layers. You can see, for example, a little pocket on the P side, a little pocket here on the Q side, which is very high identity, but is more identical to itself than anywhere else in the genome. Similarly, if you look at this more ancient material in the periphery, you can see that it's more similar to itself. And once again, this monomeric satellite, which is actually right at the edge of the centromere, actually shows that it's evolutionarily related to each other, but not any of the sequence that's in the higher order repeat structure. So this was something that was proposed basically in the 1970s, that there would be this potential mirror symmetry where the younger elements would be located in the center of the heterochromatin or the centromere. And as you move to the periphery, you would see older elements. But what is striking is really how layered and how distinct these layers are as you actually sequence and assemble. So what about centromere structure and function? Well, we know the primary role of the centromere is to help segregate chromosomes during meiosis and mitosis. And so one of the things that you get from essentially the Oxford nanopore data, which remember provided the scaffolding for sequencing across the centromere, is you get methylated and unmethylated data for CpG residues. So what I'm showing you here is that same 2 megabases, but now what I'm indicating here in red are the methylated CpGs, and in blue, the unmethylated CpGs. So as you might expect, the centromere is a hypermethylated region of the genome. So you can see it's clearly defined here. Most of the higher order satellite is methylated, and that disappears as you move to the monomeric. Perhaps surprising, but it also has been seen by Karen Miga for the X chromosome, is we find this hypomethylated block in the interior, about 76 kilobases in size. And so there's been some suggestion that perhaps this region of hypomethylation actually corresponds to the site of the kinetochore. So in the paper that will appear this week, Glenn has actually investigated this further. So this is this hypomethylated region. This is the centromere with the higher order structure now indicated here with different higher order units indicated in color. You can see there's the hypomethylated region, and then this is SEMP-A ChIP-seq data showing you a broad, essentially enrichment of SEMP-A sequence data coming specifically from this region, centered on this region of hypomethylation. And below this is essentially chromatin in situ, staining with both 5-methyl-C. You can see that most of this centromere is staining SEMP-A, which stains this specific region, and then a merge of these, which indicates pretty clearly that this region likely corresponds to the site of the active kinetochore binding in chromosome 8. So the other thing, which is mentioned by Marta, was our interest in the evolution. And so once we had the ability to sequence one human chromosome centromere, chromosome 8, we then applied it, I think for the first time, to completely sequence and assemble the chimpanzee. This is showing you the structure. And just to keep in mind the comparison with human was about 2 megabases in size. The chimp, at least this particular haplotype, is smaller. It's 1.4 megabases in size. But once again, I hope you can clearly see that there are regions that are high identity, particularly high identity, but the actual layered structure of the chimpanzee is in fact more layered than what we saw for human. So there's at least 4 or 5 layers that we can clearly distinguish, suggesting that the centromere is evolving from the center with the higher order units pushing older sequences out to the periphery, and that this sequence located at the farthest periphery, which doesn't have higher order structure, is in fact itself probably remnants of the ancestral centromere that was present millions of years ago, both in humans and as well as chimps. So we applied the same strategy to actually sequence the orangutan centromere. So this is showing you, the orangutan. You can see this here. I'm sorry, this is coming off screen a little bit. Let me try this. So once again, this is a completely different organization for the chromosome 8 centromere in orangutan. So orangutan diverged from humans about 12 to 14 million years ago. You see the actual divergent sequences at the periphery, but just keep in mind the scale. This centromere is now almost double the size of what we see in humans. We see clearly this pattern of layers once again. So this layer of divergent monomeric sequences at the periphery, then followed by sequence located here, which is obviously another layer. You can see this layer indicated here, but it's very high identity, and it's shared at like 90% or 95% with each other. But interestingly, in the interior of the orangutan centromere, there are two what appears to be very distinct higher order structures, which are indicated here by the red and the orange. But these share basically very little relationship to each other. They're not similar to each other, and in fact, even more distantly related to the sequence at the periphery, suggesting to us that this centromere may be, in fact, having evolved two higher order structures that may be, in fact, fighting for dominance. And then lastly, here is representing roughly 25 million years ago. So this centromere is now on the order of about 11 megabases in size for chromosome 8 in macaque. Once again, you can see the divergent peripheral sequences. There's, in fact, two layers of divergent sequences of monomeric satellite, which are indicated here. But instead of having the higher order structure, it has really a simpler structure of dimeric satellites, which are, as you're moving toward the periphery, are decaying more and more and showing greater divergence as they kind of move out. So putting this all together, we've now successfully sequenced and assembled the chromosome 8 centromere, not only from human, but also from two haplotypes from chimp. So I'm actually showing you two different haplotypes now from chimpanzee. I'm showing you the orangutan and the macaque. Once you've actually built these centromeres out, you can actually extract the satellite sequences and build a phylogenetic tree. So this is showing you the phylogenetic tree based on this chromosome 8 centromere analysis. Everything that you see in blue, black, and green represents essentially apes. So that includes humans. Everything indicated here in yellow or orange represents the macaque, which diverged 25 million years ago. So what we can see from this phylogeny is two, I think, very interesting things. There is clearly two different paths to the evolution of centromeres. The one that led to the formation of higher order structures in the great apes. And this is related to a subset of macaque dimeric and monomeric subunits that are present at the periphery. So this is like a sequence that is now vestigial in the macaque, but is more closely related to the higher order structures that gave rise to modern day apes and humans. And then you have on the flip side, the alternate trajectory, which led to the formation of the macaque dimeric units, which are extremely large in their formation. And there's a subset of sequences, both in human and chimp, that map to the Q arm in the monomeric satellite layer that is more similarly related to this, suggesting that there were two paths to the evolution of centromeres in the last 25 million years. So, kind of in closing, I want to just leave you with a couple of, I think, really exciting areas of future development. We now have been able to apply this phased genome assembly of trios now to essentially humans. So in terms of actually going back and now looking at humans, this is actually an assembly of the Puerto Rican centromere from a diploid source, showing you essentially the paternal haplotype fully sequence resolved, the layers numbered 1 through 5 that are present. And then this is the maternal haplotype that is shown. And when you look at these two, I think you can clearly see that they are very similar to the presentation that I gave for the other centromere of chromosome 8 from the Heterocromatin, but there are differences. So notice, for example, that this high identity region, region 5, is much larger in the mother than it is in the father. But if you look at the periphery, remember there was this other layer, we call it layer 3, and you can see that, in fact, it's essentially larger in the paternal when compared to the mother. So this is particularly exciting, I think, because we now can actually compare these sequences to each other. So shown here is the first reference chromosome 8 centromere compared to essentially one of the two haplotypes from the Puerto Rican. And you can see that the red here indicates that the sequence identity is perfect, nearly perfect identical, as you'd expect, 1 every 1,300 base pairs. But as you move to the actual centromeric satellite structures that are higher order, they become very variable, with different patterns and different patterns of conservation being seen depending upon which haplotypes you look at. So we're particularly excited by this because this means, I think, that we're going to have the ability to estimate the rate of de novo mutation in these regions for the first time. And then lastly, I'm just going to mention that we're actually redoing this analysis really for multiple human centromeres. And so this is our first attempts at reconstructing chromosome 10, so fully sequenced assembled chromosome 10 from the T to T assembly. and then using our ONT high 5-base strategy to build out the chromosome 10 corresponding centromere from orangutan. And I know this is a lot to digest. I don't know how much of this you guys can see at your end, but this is the view from the telomere to telomere consortium of all human centromeres now fully sequenced and assembled. I think we're missing Y, but Y has already been assembled previously. This is showing you that patterns that I showed you before. I'm indicating actually SEMPE binding as well as the hypomethylation. And I think you can clearly see that most centromeres have this hypomethylated dip, which corresponds to regions of centromere essentially binding, suggesting that we're actually now identifying the specific kinetochore binding sites within centromeres for the first time. Okay, so with that, I just want to leave a few thoughts in closing. The first thing I want to mention is that sequencing 32 human genomes is not enough. To completely understand the complexity of human genetic diversity, we're going to need to sequence many more. And there's a major initiative underway across the world, actually, but there's a lot of it being done right now in the US. It's called the Human Pangenome Project. And the idea is essentially to build over the next few years telomere to telomere assemblies of fully phased human genomes, sampling roughly around 350. And that's probably not even going to be enough to fully understand the organization. In my lab, as already mentioned, we're doing a similar effort in addition to doing multiple humans. We're also attempting to do the first telomere to telomere assemblies of non-human primates. There are 448 distinct species of primates. We're focusing, shown here in the boxes, on genomes that in fact represent index nodes for essentially interpreting the human evolution. So we are planning to sequence, and already have sequenced close to about a dozen individuals across this primate phylogeny tree. And we're now working on actually finishing those bases across the difficult regions, really to understand the evolutionary history of every base of the human genome. I can tell you that we're starting to get new insights in terms of mutational mechanism. And so a paper that we recently published with David Perupsky looked at the pattern of inversions across the human genome. So this is something I haven't talked much about. We're focused on insertions and deletions and duplications, but there also are regions of complex inversion polymorphism in the human population. And what I'm showing you here are snapshots of a few megabases in size, where each line here represents either a non-human ape haplotype or a human haplotype, which is listed here as HG all the way down to NA. And all I'm indicating here is essentially the orientation of the sequence, whether the sequence is in a forward orientation, like the reference genome shown here in green, or in reverse orientation, i.e. an inversion shown here in orange. And maybe not a surprise to some of you, but there is in fact a complex pattern of inversion polymorphism extending over megabases of the human genome, where the order of the segment is actually flipped in some individuals. So look at this region here on 16p12. It's a very gene-rich region. It's actually in 1 out of about 40 individuals that we've characterized, the orientation is flipped, largely as a result of unequal crossing over between those segmental duplications. This is not an exception. It is, in fact, seems to be a rule for humans. And in fact, this flipping back and forth, which we call toggling, persists among the apes. So these regions of the human genome have been flipping back and forth probably for tens of millions of years. And then last, I just want to mention that obviously one of the applications of this long-read technology will be to characterize patients. And so this is some work we did with a former postdoc of mine, Heather Mefford, who discovered one of the common causes of Baratella-Scott syndrome. So this is a pediatric disorder, which is caused by loss of a gene called XYLT1 in a recessive stage. But it turns out that a lot of patients that actually have this disease don't actually have a standard mutation, but instead have a triplet repeat expansion mutation in a CGG repeat located in the 5' end of the XYLT1 gene. So shown here is a patient who has actually was unlucky enough to actually have an expanded allele in both the paternal and maternal complements. So this patient developed Baratella-Scott. This is how this was discovered about 3 years ago by Heather Mefford and her postdoc. If we take that same patient and actually sequence it, sequence the mother as well as the proband, you can clearly see the fully phased and assembled CGG repeat expansion that is present. In fact, we can also infer methylation status of this if we actually have the Oxford nanopore data. So this is a clinical fellow in my lab that has actually now been targeting unsolved cases of disease. In this particular case, he's been working on Mendelian causes of disease. And so this is a patient that was known to have a complex rearrangement by cytogenetics, shown here at the bottom. It was a karyotype workup. You can apply a technology called targeted long-read sequencing, also referred to as read-until with Oxford nanopore, where you select the region that you want to characterize, and you just sequence molecules coming from that specific region. So you throw out 99% of the genome and you focus on that. And when Danny did this, and I'm not going to go through with that, he was able to actually refine every breakpoint in this patient, discovering new breakpoints which are actually have clinical consequence in terms of the management of this patient. I think it's clear to me that the cost of this type of targeted long-read sequencing is actually well within the range of what we can now afford with next-generation sequencing. And I think this is going to be an immediate application to improving clinical care of patients. So in summary, a few take-home messages that I would like you to keep in mind. Long-read sequencing is, I think, revolutionizing the way we study human genetic variation, because fundamentally we no longer are aligning sequence reads to discover variants, but we're actually building assemblies. And by building assemblies, we actually put the variation into complete context. So just to remind you that compared to Illumina whole genome sequencing, 75% of the structural variation that's detected by long reads is essentially missed by Illumina. And about 95% of the structural variation is missed by applying strictly bio-nanogenomics. And that's because the long read sequencing is getting those smaller events that are being missed. The second major point is combining hi-fi sequencing data and linking read technology, such as StrandSeq or Hi-C, allows us to phase genomes. So we now can distinguish the maternal versus the paternal assemblies. And this actually allows us to actually put those structural variants into context of flanking SNPs, which allows us to improve genotyping. Combining both Oxford nanopore and hi-fi sequencing, we think is the future for actually solving the complex regions, such as heterocromatin, such as segmental duplications, because it's providing us access to these regions for the first time. So for those of you interested in Robertsonian translocations or interested in essentially actions that are going on in the centromeres, there's huge numbers of chromosomal abnormalities related to ring chromosomes and paracentric inversions and so on. This combination of long reads, both Oxford nanopore and hi-fi, is a way to actually resolve these for the first time at the sequence level. And then lastly, if I live long enough, I believe this will become the way we sequence and assemble genomes clinically for the discovery of pathogenic variation. So this is my group. I want to just really acknowledge them for still being very active during this very difficult time of COVID. And I also want to acknowledge all the collaborators. The people that are highlighted here allowed me to present unpublished work at this meeting. But I also want to thank my great colleagues as part of the Human Genome Structural Variation Consortium, Tobias Marshall and Peter Abert. My colleagues from the Telomere to Telomere Consortium, Adam Philippi, Karen Miga, Serge Korn, and Sergey Nurk. These are guys that are computational scientists that have developed some of the first algorithms for sequence and assembly. And then colleagues really from around the world that have been critical to some of the work that I presented today. And with that, I think I'm right on time. I'd be happy to take any questions if we have time. Thanks.
Thank you so much, dear Evan, for your wonderful talk. Your chat on YouTube is full of messages about your lecture. It's a really, really, really great success. So we have some questions. We try select a few of them, and I'd like to invite Dr. Peixoto and Dr. Marta to help me in these discussions. Please, Dr. Peixoto.
Hi, may I say it was quite interesting. Thank you so much. It was excellent.
Oh, thank you so much.
Yeah,
I would have much rather have been visiting you guys in Brazil as opposed to doing this with my dog crying in the background. But anyways, I hope it wasn't too much of a distraction.
I will make sure that if we get through the pandemic, you will come to visit us one day.
For sure.
That's excellent talk. I will start with the comment from Dr. Claudia Carvalho here, and she said, terrific presentation. Thank you, Dr. Eichler, and the organizers. So
well,
I
thank
think
you, Claudia.
with her,
yeah, I know her from many years, yeah.
Yeah, I think with her note, gives you a little sense of the wonders that you just caused in everybody who is attending this conference today. And I will start with two questions from hold on for a second. Gustavo Kahn. First question, he asks you, why are the young set DNA repeats in humans restricted to the core of centromeres? And the second question. Okay, go ahead.
Yeah. So it's a good question, because you obviously, and if you remember me showing you the orangutan, it was different there, right? There were two, and it wasn't clearly the center. I will make this point. When we look at other human centromeres, I only showed you chromosome 8 in detail. Not every human chromosome has the high identity at the center. Some of them have it shifted toward the right or the left, so more P versus Q. Some have multiples like the orangutan. So this may be just a chance phenomena that that's occasionally it occurs right at the center, creating this perfect mirror symmetry. In other cases, it's shifted and it's asymmetrical to the left and to the right. And so I think when we actually sequence many more human haplotypes, and we sequence many more non-human apes, we'll see this diversity of shifting to the left and to the right, which might have something to do with what it takes to make the most competent centromere. There's a lot of hypotheses out there, and I didn't go into detail in this, but one thing I will tell you, remember I showed you the high identity being at the center, right, of that chromosome 8? The actual kinetochore binding wasn't over that site. It was shifted to the right of that. So the hypermethylation dip, where the CEMP-A binds, is actually not at the most highly identical region. And this is actually goes back to some old theory that says that there's a constant kind of arms race in the centromere to produce the most competent kinetochore or centromere binding site. And sometimes mixing higher order repeats together creates a more competent one. And that's clearly what we're seeing in CIN8. And so I think I think one of the things that will be really exciting is seeing how the pattern of the high identity segments versus essentially the kinetochore binding sites shifts for different humans, for different species over time. Because I have a feeling it's very plastic, and it's constant. You know, we don't know this yet, but it's possible that the kinetochore binding may be different in different human haplotypes at different positions. But it's a great question, and I wish I had more of an answer, other than to tell you that it's not seen in every human centromere. It's a subset. It's about a third of the centromeres that show this mirror symmetry that we see.
I think one great observation I have from your talk, it's about the plasticity of the chromosomes. The early assembles and the early genomic reference were kind of stuck in the things that were quite common and we were seen as a functional genome. However, your work shows how plastic it is. Now, yeah. It's a question of mine. Do you have a hunch? I know it's too early for it, but to favor one of those two theories about the evolution of those centromeres, because I clearly think that one of the reasons that we see the difference, that's more than two mechanisms may be involved in the production of the variants within that region. So do you have a hunch for which mechanism may account for that?
Yeah, I think the mechanism for diversity, for kind of, if you think about generation of more diversity in centromeres, which are much more mutable than most regions of the genomes, is separate in a way from functionality. So I think the functionality happens, but the formation of where the kinetochore occurs in the centromeric heterochromatin is essentially dictated by a number of different phenomena that occur. And so while the generation of diversity is valuable in allowing you to test out new centromeric combinations of sequence, right, that may be useful, it's the diversity itself operates independently from the function. And the reason I say this, and it's some of this is unpublished and some of this will appear in the paper, we also characterized a site of neocentromere formation, which, as most of you know, neocentromeres are things that typically occur outside of alpha satellite. And what we found, interestingly enough, was a completely different set of repeat sequences that had expanded. And we actually went back to the neocentromere cell line. So this isn't part of the paper, but this is the work of Glennis. She went back to the cell line. And in that cell line, where the neocentromere had taken over from the normal centromere, the sequence of this new repeat had expanded to megabases in size, almost suggesting that this became more competent than the centromere itself, and then co-opted and it led to the formation of a new centromere in this one human cell line. So the really cool thing, when she was showing me this data and she was like mapping the SEMP-A data on top of it and so on, I was like, wow, you're actually seeing evolution in action, like in real time. Right? You're seeing a shift away from satellite. So while the satellite will continue to diverge and do its dance, it could be that a new sequence will take over and become the predominant one, right? And it goes to some old evolutionary theory about centromere repositioning, how centromeres constantly are repositioning over evolutionary time, you know, in the same vicinity, but different locations, right?
Right.
You know, they're never happy with their first apartment, right? They have to find a better apartment.
Yeah, I could stay here all day just talking to you, but I can't monopolize you. But I think you took a picture of evolution. I don't know if it was happening or had happened and was reformulating when that, I think on chromosome 8, you showed that there are two flanks alongside the arms of the chromosome that shows a little bit of old centromeres or perhaps a new centromere information there. But anyway, so Gustavo Kahn said, what could be the advantage for centromere structure and function having HOR instead monomeric at the core of the centromeres? Do the hypomethylated regions match to the young repeats?
Yes, so the answer is no, they don't. They're not the youngest. They are young, but they're not the youngest repeats. So that was what we were kind of mentioning before. There is the whole region is higher order structure, but then there is higher order structure that's 100% identical, and then higher order structure that's 99% identical, 98% identical. Where the kinetochore bound was actually an area where it was roughly 97 to 98% identity in terms of the higher order structure. But what was interesting, which we didn't talk about, but is mentioned in the paper, is that it's an area of increased entropy in the sense that there's more diversity of higher order structure at that location. So it's not homogeneous, but it's actually heterogeneous higher order structure, which I think is very informative because it suggests that the different higher orders all have added some value, and that's the preferred site for kinetochore formation, at least in that cell line.
I think now Dr. Marta will present you another question. Thank you.
Yeah,
Okay.
there are many
Hey,
questions.
Marta.
Hi. So we have a question from Isabel Almeida, and she would like to know if you had any interesting findings on the Y chromosome.
Well, so I can tell you, to be honest, not a lot yet. The actual structure on the Y is in fact very simple in a way compared. It is very small, as some of you know, in terms of the centromere. And in fact, as so far from what we've looked at, and again, we haven't looked at many human haplotypes yet, it doesn't show as much variability. But again, I mean, our numbers of haplotypes that we've sequenced and assembled are relatively few at this point. The short answer is we don't know enough yet to say.
Yeah, the Y chromosome is always like, how do you say that? The black sheep? No.
Yeah, it is
the
black sheep.
It's the different one always. And Luciana Lorenzo asked, is there any relationship between the different layers of centromere repeats and their three-dimensional positioning in relation to the kinetochore?
Well, that's a great question. Short answer there is we don't know yet. Really, we just got this data literally in the last 3 or 4 months, right? But it's a great experiment, and I'm sure Glenys would be interested in talking to you guys more about this. For those of you who don't know Glenys, she's a true centromere biologist. She came to my lab kind of when we were applying these new sequencing technologies, and she said, I want to do this, and I want to study. I'm not interested in just structure. I want function. And so she's very interested in these questions that she just raised, right?
Is she only interested in human centromeres or centromeres in general?
Well, I'm getting her more and more interested. I got her outside of human to do the chimpanzee and the orangutan.
Yeah, but I'm thinking about marsupials.
For sure. I'm sure she could be convinced, right? I've already got her thinking about apes, right?
Yeah. They are at the end of the evolutionary. And I worked on marsupials, and you can tell their genome has many differences from other mammals. That's why I was thinking.
Interesting.
Anyway, we'll have to talk a lot about it later. Claudia Carvalho said, Dr. Eichler, I found fascinating that acrocentric chromosomes have the P arm laden with segmental duplications. Are Robertsonian translocations of chromosomes 13 and 14 mainly due to non-allelic homologous recombination?
Yeah, this would be a question exactly like Claudia would ask. We don't know the answer to that, but it's a great hypothesis, and I think this is something I actually talked to. Some of you may know a person by the name of Stylianos Antonarakis from Geneva. He's been very interested in acrocentrics and Robertsonian translocations, and I asked him, how well are the breakpoints mapped for Robertsonian translocations? And he said, in a few cases, they're very good, but in most cases, it's not very well known. So, Claudia, I think it's a great hypothesis, right? It could be that the breakpoints are not happening in the satellite. They're not happening in the rDNA, which is what most people think. It could be that the breakpoints are actually mapping in the sea of segmental duplication, which are 99, 98% identical. The one thing relevant to this that I think is important, when we've mapped the length of segmental duplications across the genome, the longest segmental duplications exist in the acrocentrics. In other words, the process that homogenizes them creates longer, more identical seg dupes than we see anywhere else in the human genome. So I think it's a safe bet, but it hasn't, to my knowledge, been proven that those are actually mediating some of the Robertsonian translocations.
I have a question regarding those chromosomes. Outside the centromere, you also showed that they are very poor in genetic material. We know that for sure. And you also showed today that they are enriched in segmental duplications. Some more than others, but most of them are all enriched in segmental duplication. Isn't that favoring a way to show that evolution is actually removing genes from those chromosomes and placing them elsewhere? And in the long run, we are going to lose the functionality of those chromosomes altogether.
Actually, I think of it the other way.
You do?
I think because they are receivers, not senders, in the most part. In other words, the segmental duplications that are going off to the acrocentrics, they have counterparts on the normal chromosomes. So they're picking up material, and that actually churning that material around.
Yeah, they are deposit.
Yeah, so years ago we studied in our lab, for example, it was almost 20 years ago, we studied paracentromeric regions of chromosomes. And there we found that there were a lot of gene bodies, quote unquote, duplicated, but rarely functional genes formed. But occasionally a new gene emerged, right? And so there was an old colleague of mine who's now retired named Barb Trask. Some of you may remember Barb. But she saw the exact same phenomenon at the ends of the chromosome. So now that we see these acrocentric sequence resolved, they remind me of the paracentromeric and subtelomeric duplications that we see in these other areas of the genome, where they're constantly bringing in new material, but it's like a churn. It's like it's constantly being churned like a blender. To your point, we don't see a lot of evidence of transcription yet, but we may not be doing this the experiment the right way. We might need to look at other types of RNA polymerase data sets to actually characterize what the transcriptional landscape looks like. It may not be polyadenylated material. It may be other things. But now that we have the reference, we have our reference, right? And we know it's very variable. We can start to really identify these segments that are fixed versus variable. We can characterize the seg-dubs. We can characterize the genes, the content, everything, right? I think it's a fun time for students. I tell students, man, I wish I could turn the clock 20 years, back. And I was a student right now, right? This would be a fun time to be getting access to all this data.
Yeah, sure. It's good to be alive.
Yeah. Can I ask, there's another question from a good friend of mine, Anna Kripiski. Thank you for your great talk. Do you think that specific centromere structures could drive somatic mutations impacting cancer predisposition, mainly in pediatric cancer enriched in SP?
Yeah, that's a great question too. I do. So the short answer there is I do believe that there's a phenomena there. I mean, the limited I know about somatic mutation associated with cancer, right, is it's obviously highly selected. It's related to methylation often. Methylation changes in culture. And it's not the same as what we and Claudia see in terms of non-allelic autologous recombination. It's a different phenomena. I do know that certain cancers actually show biases in terms of the types of rearrangements. So certain cancers, I remember from years ago reading sarcomas in particular, show this pattern near centromeric regions or pericentric meric regions. So I think it depends upon what kind of cancer you're looking at in terms of the types of mutational mechanisms that are occurring. But it's certainly an area worth investigating, I believe. I mean, to be honest, we just don't know how what I'm really interested in is understanding what the new mutation rate is for these regions. I mean, how rapidly do they really change? We estimate now that they're evolving 4 times more rapidly than most of the genome, but that's a lower bound. The number could be much, much higher, and somatically, we just don't have a handle yet.
Okay, I have another question from Professora Luciana Lourenço. Is there any relationship between the different layers of centromere repeats and their three-dimensional positioning in relation to the kinetochore?
Yeah, we don't know. But I think it's a great question, and I think can now be pursued. There's a young faculty named Brian Bellevaux, who is actually in my department, was very interested in these questions of three-dimensional structures in heterochromatic sequence. Now that we have the sequence and we can design probes that are specific for specific parts, it should be possible to address this.
Let me see. Ludmila. So, Ludmila Oliveira, she said, fascinating work, especially about the centromere sequencing. Do you guys know if the divergent sequences on the edges of centromeres are similar among different chromosomes? I think you talked about it.
Yeah, so they do share some similarities, but so having said that, so the answer is if you're referring to the monomeric tracks, which are at the periphery, they are more identical to themselves, right, than they are to the interior, which is the higher order. But those sequences themselves in the monomeric, although they show divergence, right, to other monomerics, there is some similarity that exists, suggesting that the ancestral centromere that would have existed probably 25 million years ago may have come from some common source itself and distributed across the centromeres. We did some early phylogenetic reconstructions almost a decade ago, looking at this question of the relationships of the monomeric. It really does appear that there are different trajectories of the evolution of the centromeres, and the monomerics represent kind of a vestigial or residual state.
Peixoto, do you have more? I think we are done with the questions from the chat, but we have plenty of questions.
I would have a full day of discussions.
Yes, so
but
the deal is
next time we can
yes,
have a.
Yeah.
I think the deal is you're definitely coming back because you
Yeah.
saw many people are interested in talking to you here.
Yeah. I would happy to come. I've only been, like I think I was telling Marta, I've been to Brazil once before, but I was really impressed with the people that I met, the scientists that I met there, and I was especially impressed with how the young people partied. But I just remind them that I'm not a young person anymore, so I don't party that way. But it was such an impressive meeting I went to. That was, I guess, almost a decade ago. But I enjoyed my visit, and I look forward when life comes back to normal. And
Yes.
God willing, it comes back to you guys as quickly as possible. I know it's been hard for Brazil. I wish you Godspeed in recovery and back to a normal life, because this has been a hard time for everyone. And it's a pleasure to meet you guys again. It's Marta, reminding me of the days we met in Florence many years ago now. And meet
3
some of the new
years.
people.
And I also
So,
acknowledge
2017.
who presented questions to you. Everybody responded here on the chat line, saying that they were satisfied with your answers, and they were grateful to hear from you, especially your friend Claudia, and also all the other professors that asked. They are all thanking you for your kindness. And me too, I'll thank you again most humbly for your time, and I appreciate every minute of our discussion. And if I may bold to ask you if I could entertain some questions through email to you, sir. Yeah.
For sure, I'd be happy to. Just let people know that I'm going to go on family vacation for the next few days, so I won't be responding for email until next week. But
Oh, no,
I'll look for your emails.
Okay, it won't be right away, but I will contact you in the near future. Thank you so much.
Excellent. Well, pleasure, guys. I'm glad that we were able to communicate across the continents.
Yeah.
Technology shortened this.
Yes, it did.
So thank you very much for your time, for this incredible lecture. A lot of people love your presentation and talking. And the reason was a really pleasure listening you today. Finalizamos aqui a nossa live de hoje e convidamos a todos para o nosso próximo encontro, que será no dia 28/4, às 14 horas. O tema é Design and Application of Oligofish. A palestra vai ser proferida pelo Dr. Jimin Jan, from Michigan State University. Novamente, muito obrigado pela presença de todos. Muito obrigado, Marta e professor Peixoto, novamente. E espero poder encontrar vocês em nossa próxima transmissão. Muito obrigado.
Obrigada.
Bye, guys. Ciao.
Evan, wait a minute.
Bye.
He's gone.
Bye, Ivan.